bacteria. Phylogenetic analysis, using 16S ribosomal RNA, revealed distinct domain of bacteria. Ribosomal RNA is present in all organisms and can be used for analyzing evolutionary relationships. The three domains of life are Domain Bacteria, Domain Archaea, and Domain Eukarya. Learning objectives. 1. Describe the general morphology and characteristics of bacteria. 2. Understand the role and importance of bacteria in the environment and industry. General morphology of bacteria. Bacteria are prokaryotes. No membrane-bound nucleus and organelles. Bacteria shares general morphology with Archaea. Cell wall. Functions as a protective layer and responsible for the cell shape. Plasma membrane. Serve as barrier and separates cell from the external environment. Cytoplasm. Composed of complex solution of organic molecules. Ribosomes. Where protein synthesis occurs. Chromosomes. Consists of circular, double-stranded DNA, contains genetic information located in the area of the cell called nucleoid. Plasmid. Consists of extrachromosomal DNA. Capsule. Allows for attachment and protects cell from dehydration and attack by phagocytic cells. Flagella. Singular flagellum used for locomotion. Pili. Singular pilus used for attachment to surfaces. Prokaryotes have three basic cell shape: cocci or spherical, bacilli or rod-shaped, and spirally or spiral-shaped. Bacterial shape is genetically and environmentally determined. Stelohumosi. Star-shaped bacteria. Marine bacteria. Haloarcula quadrata. Rectangular-shaped bacteria. Marine bacteria. Cell envelope. External covering outside the cytoplasm. Composed of two layers. Cell wall and cell membrane phospholipid bilayer. Two main groups of bacteria are classified based on the difference in cell wall. Gram-positive bacteria. Thick peptidoglycan and cell membrane. Gram-negative bacteria. Outer cell membrane, which contains lipopolysaccharides or LPS. Thin peptidoglycan layer and cell membrane. Phospholipid bilayer. Two layers of phospholipids, which surrounds the cell, hydrophilic exterior, water-loving, and hydrophobic interior, water-hating. Ester-linked lipids. Endospores. Some gram-positive bacteria produce endospores sporulation, which are highly resistant to extreme temperatures and chemicals. Endospores are inactive resting stage of bacterial cells capable of returning to vegetative growth, germination, in favorable environments. Gram-positive genera, which form endospores. Bacillus, Clostridium, Sporosarcina. Flagellar arrangements. A. Paratrichus. Flagella dispersed over the surface of the cell. B. Monotrichus. Single flagellum at one end. C. Lophotrichus. Small bunches at one end. D. Amphitrichus. Flagella at both ends. Modes of nutrition. Autotrophs. Photoautotrophs. Derives energy from light. Purple sulfur bacteria, cyanobacteria. Chemoautotrophs. Derives energy from inorganic substances. Heterotrophs, or chemoorganotroph. Parasite. Lives and feeds in a living host, which may cause disease or illness. Saprobe. Decomposers that feeds on dead organic matter. Modes of reproduction. Asexual reproduction through binary fission. Binary fission. Parent cell divides into two identical daughter cells. Horizontal gene transfer, HGT. Transmission of genetic information on the same generation and not from parents to offspring. Transformation. Uptake of DNA genetic material from external environment. Conjugation. Transfer of DNA through direct cell-to-cell contact mediated by conjugation pilus. Transduction. Transfer of DNA mediated by a virus particle bacteriophage. Habitat and ecology. Lives everywhere, even in extreme environments. Temperature adaptation. Psychrophiles. Thrives in temperature between 0 degrees Celsius to 20 degrees Celsius. Mesophiles. Thrives in temperature between 10 degrees Celsius to 50 degrees Celsius. Thermophiles. Thrives in temperature between 45 degrees Celsius to 80 degrees Celsius. Oxygen adaptation. Obligate aerobes. Grows in normal atmospheric oxygen and handle toxic oxygen byproducts. Obligate anaerobes. Can live in environment without oxygen, killed by oxygen. Facultative anaerobes. Aerobes, which can live without oxygen. Aerotolerant anaerobe. Cannot use oxygen for respiration and not killed by oxygen. Microaeropile. Aerobes, which requires small amount of oxygen but does not grow under anaerobic conditions. pH adaptation. Acidophile. Thrives under acidic environments below pH 7. Neutrophile. Grows best around pH 7. Alkalophile. Thrives under basic environments above pH 7. Pressure. Barophiles. Adapted under highly pressurized environment, bottom dwellers in the ocean. Involved in biogeochemical cycles. Nitrogen fixation, sulfur cycle, carbon cycle. Microbial taxes. Bacteria responds to stimuli from the environment. Chemotaxis. Response to chemicals. Chemoattractant. Attracts bacterial cells. Chemorepellent. Repels bacterial cells. Phototaxis. Response to light. Aerotaxis. Response to oxygen. Osmotaxis. Response to ionic strength. Hydrotaxis. Response to water. Magnetotaxis. Response to magnetic field. Scotophobotaxis. Response to changes in illumination. Representative bacteria. Bacillus subtilis. Gram-positive bacteria. Rod-shaped cells, bacillus. Endospore forming. Paratrichus flagella. Staphylococcus aureus. Gram-positive bacteria. Spherical-shaped coccus. Non-motile. Non-spore forming. Produce staphylococcal toxins which cause food poisoning. Escherichia coli. Gram-negative bacteria. Non-spore forming. Paratrichus flagella. Cause food poisoning. Used as competent cell in recombinant DNA technology, genetic engineering. Spiral involutions. Gram-negative bacteria. Spiral-shaped cells. Amphitrichus flagella. Cyanobacteria. also called blue-green algae, gram-negative bacteria, aquatic and photosynthetic, nostoc, oscillatoria, spirulina, involved in carbon fixation. Products from bacteria: Streptomyces sp. Antibiotics: streptomycin, tetracycline, erythromycin, lactobacillus sp. Lactic acid, Clostridium acidobutylicum, acetone, rubbing alcohol, Bacillus sp. Microbial plastic polyhydroxybutyrate, or PHB.